Bismillahirrahmanirrahim. In our last lectures, we discussed the cell-derived chemical mediators, and today we will discuss the uh, plasma-derived chemical mediators of inflammation. Now, there are three groups of plasma-derived chemical mediators of inflammation known as the complement system, kinase system, and clotting system. And in plasma derived chemical mediators of inflammation, uh, we must know that actually most of these proteins produced in the liver and also in the cells outside the liver, but they circulate in the blood in their inactive form. And once they are activated, they play their role in inflammation and immune systems. The three plasma derived chemical mediators, the complement system, the kinase system, and the coagulation system. These are proteins, as we know that most of them are synthesized in the liver, but some proteins they are also synthesized by the cells outside the liver, and they circulate in the blood in their inactive form. Now, what triggers them to become active? The microbial components, particularly the microbial polysaccharides, microbial membrane components, and the antigen antibody complexes, and some of the blood coagulation components like the Hagman factor or factor 12 and some cytokines, so, which are derived from the cells, mostly the inflammatory cells. So they will trigger these inactive proteins and get them activated, and after they have got activated, they will play their role in inflammation and also in the immune system. Now, the first component of the plasma derived chemical mediators is the complement system. The complement system actually consists of 9 complement proteins from C1 to C9. And what happens that once a protein of the complement system is activated, this activated protein will activate and break down other proteins into their active components. For example, the three pathways that are involved in activation of the complement system, the classical pathway in which the antigen antibody complex activates the complement 1 protein. Now, the complement 1 protein then cleaves the two proteins complement 2 and complement 4. The C2 is broken into C2A and C2B. Similarly, the C4 is broken into C4B and C4A. Now, one component from the C2 known the C2B and one component from the C4 which is C4B. These two combine to form the C3 convertase. This C3 convertase will break down C3 into C3A and C3B, and all of these components have got their roles in inflammation. Now, the C3B, which is formed by the breakdown of the C3, along with C3 convertase, it will form the C5 convertase, and C5 convertase will break down the C5 component into C5A and C5B. Now, there are three pathways for the activation of the complement proteins. Number one is the classical pathway, which is triggered by the antigen antibody complexes, and they start from the activation of the C3 convertase. The alternate pathway, which is activated by the bacterial components like bacterial wall components and polysaccharides, and the third pathway is the lectin pathway, in which the lectin which circulates in the plasma, it's an active form, it binds to the carbohydrate component mannose and the bacteria, and all the three pathways ultimately leads to the activation of the C3 convertase. Now, what happens here that the C3 convertase will be break C3 into two components, C3b and C3a. Now, the C3a and C3b, which are derived from the breakdown of the C3 by the C3 convertase. Now the C3b along with the C3 convertase will form the C5 convertase, and C5 convertase will cause the breakdown of the C5 into its two components, C5a and C5b. Here we can see that the components of the complement system, particularly the C3A and C5A, they are strong anaphylatoxins, and they act just like in type 1 hypersensitivity reactions, which is also known as the anaphylactic reaction, or in which what happens that in type 1 hypersensitivity reaction, the antigen antibody complexes, particularly the immunoglobulin E, cause breakdown of the mast cells. So similarly, the C3A and C5A, they can break down the mast cell and cause the release of histamine. And we know that histamine is a strong vasodilator, as well as it increases the vascular permeability. The increased caliber of the blood vessels, which is caused by the histamine known as the vasodilation, and the increased vascular permeability, in which the tissue, the fluid from the blood and as well as the inflammatory cells from the blood, they go into the extravascular tissue, and they are responsible for the cardinal signs of inflammation, like erythema and warmth, in which the tissue, inflamed tissue, becomes hot, feels hot, and exudation, which is due to increased vascular permeability, which result in the edema and swelling. In addition, C5A, it is a strong chemotoxic agent, particularly for the neutrophils, and it causes the release of inflammatory mediators from the other cells. Now, C5, which is broken down by the C5 convertase into C5A and C5B. Now, this C5B, along with the C6, C7, C8, and C9, it forms the membrane attack complex. And this membrane attack complex forms holes, punches in the membranes of the bacteria and kill them. Out of these complement components, the C3b, which is formed by the breakdown of C3 by the C3 convertase, it is a strong opsonine. Normally, our immune cells, particularly the phagocytes, they do not recognize the bacterium. But if C3b is attached to this bacterium, they can recognize the C3b, and in this way, the phagocytes, they have got receptors for the C3b, and the C3b which is attached to the bacteria, these phagocytes will attack those bacteria and will kill them. Now, in addition to the A and B forms, the complement proteins also have an active and active form. And the three pathways, the classical pathway, which is rapid and rapidly acting, and the alternative pathway and lectin pathway that are a little but slowly acting, they form and attack the bacteria through the membrane attack complex, as well as through C3b, which acts as an opsonine, and they are involved in their effects on the blood vessels, leukocytes, and phagocytosis, and the membrane, through the membrane attack complex, they can cause direct killing of the bacteria. Now, in this slide, we can see that the A and B components of C3, C4, and C5 are actively involved in inflammation and immune reactions. For example, the C3A and C3 and C5A, these are strong anaphylatoxins, and they cause breakdown of the mast cells and cause the release of histamine, and which causes strong vasodilation and increased blood flow, and they also increase the, particularly the C5A, increase the permeability of the blood vessels, and in this way the inflammatory cells and fluid leave the blood and go into the extravascular space. They are also involved in leukocyte activation, adhesion, and chemotaxis. The C3b, as we said, that it is involved in opsonization, which is the process in which the opsonin binds to the bacterium and make it target for the phagocyte. The C5b, which is results from the breakdown of the C5 by the C5 convertase, this C5b along with the C6, C7, C8 and C9, it forms the membrane attack complex which form holes and punches in the bacterial wall and kill them. Now here in this slide we can see the C5a and C3a. They are involved actively in inflammation and are involved in the recruitment and activation of leukocytes and destruction of the microbes by the leukocytes through opsonization and phagocytosis. The C3b, which acts as an opsonin, is involved in phagocytosis of the bacteria. The membrane attack complex form holes in the wall of the bacteria and cause its destruction. Now, here we summarize the functions of the complement system. C3 and C5a, they are strong anapylotoxins and they increase the vascular permeability also and cause vasodilation. And uh, this function is achieved by the breakdown of the mast cells which release histamine. And C5a also activates the lipoxygenase pathway. If you remember, during the arachidonic acid metabolites, we have studied that there are two pathways, the cyclooxygenase pathway and the lipoxygenase pathway. The cyclooxygenase pathway results in the formation of prostaglandins and thromboxane A2, while the lipoxygenase pathway results in the formation of leukotrienes and lipoxins. The C5a and C3a and C4a, they activate leukocytes and play their role in inflammation by increasing the adhesion of the leukocytes to the endothelium. And C5a itself is a potent chemotoxic agent. It induces chemotoxis in which it uh, induces the directional movement of the um, inflammatory cell toward the attacking agent or bacterium. IC3b, which is a precursor form of the C3b, along with the C3b, they act as strong opsonins. And C5b to C9, they form the membrane attack complex. Now, here we conclude that C3IB and C3B, they are important opsonins. The C3A and C5A, they are strong anapylotoxins and cause the mast cells to release histamine and to cause vasodilation and increase vascular permeability. And additionally, C5A is a strong chemotoxic agent as well as it causes the release of inflammatory mediators from the other cells. Now, this complement system should be strictly regulated because if it is left active, it will cause severe injury to the healthy tissues. As we know that inflammation itself is a double-edged sword. The basic purpose of inflammation is to provide protection to the body, to provide protection to the healthy tissues, to kill bacteria, to kill viruses, to kill fungi and parasites. But if inappropriately activated, it may cause damage to the uh, body healthy tissues. So there are regulatory mechanisms that only allow the complement protein activation uh, in a limited way, and there is when the activation is not needed, they stop them and decay them. So if there is a problem in this regulatory mechanism of the complement system and in this check and balance, what will happen that the inappropriately activated complement system will damage the normal cells. And there are certain diseases that result from the unneeded or not required or inappropriate activation of the complement system, and these diseases are the result of some problems in the regulatory system in which the complement system is not properly regulated, and there may be some inherited deficiency of some proteins that are involved in regulatory system. And what happens that the inappropriate activation of the complement system will result in the diseases like hereditary angioedema, hemolytic uremic syndrome, macular degeneration of the eye, and paroxysmal nocturnal hemoglobinuria. In this example, we see that there is a regulatory protein which is known as the C1 inhibitor, and it will control the activation of C1. It will inhibit the activation of C1. And this C1, when activated, it will cause the kinine system conversion of the pre-kallikrein by the Hagman factor into kallikrein. And this kallikrein will convert the high molecular weight kininogen into bradykinin. And bradykinin itself uh, is a strong component which is involved in pain, fever, and other conditions and sign symptoms that result from inflammation. So in this example, in hereditary angioedema, we see that genetically if the person is deficient in the complement 1 inhibitor, and if there is no complement 1 inhibitor, or if it is defective, what will happen that there will be excessive activation of the uh, kallikrein, and it will form lot of bradykinin which will cause damage to the healthy body tissues, and the damage may be in multiple tissues, particularly including the larynx, which may lead to severe respiratory problems. Now, in addition to the complement system, the three other systems are the kinine system, clotting system, and fibrinolytic system. Now, role of the clotting system, we discuss that actually the Hagman factor, which if you remember we have studied in hemodynamics, and it is involved in the intrinsic pathway of coagulation. And what happens that in the intrinsic pathway of coagulation, when this inactive Hagman factor or factor 12 touches the collagen basement membrane, or if it comes in contact with the activated platelets at the site of endothelial injury, or at the site of inflammation, or at the site of injury, so it becomes activated. And once it becomes activated, it will convert the pre-kallikrein into kallikrein. And the kallikrein will convert high molecular weight kininogen into bradykinin. And this bradykinin will cause increased vascular permeability, vasodilation, particularly the arteriolar dilation, and it is very dangerous in asthma and asthmatic patients because it causes bronchial smooth muscle contraction, and it is also responsible for the pain that we suffer in infection and inflammatory processes. But this bradykinin should be quickly degraded by the enzymes known as dekinases, which are present in the plasma and tissue. And if this bradykinin is not rapidly destroyed, or if its formation is excessive, that what we see in hereditary angioedema, in which the C1 inhibitor protein is missing, what will happen that there will be excessive uncontrolled formation of the bradykinin, and it will cause damage to the body and healthy body tissues. Now, after the coagulation system, in which the factor 12 or Hagman factor is actively involved in the plasma derived mediators of inflammation, here we can see that the fibrinolytic system is actually involved in the degradation of the uh, fibrinogen into fibrin, and then degradation of the fibrin by the plasmin. So in this slide, we can see that the Hagman factor or factor 12, which cause the formation of thrombin from the prothrombin in the intrinsic pathway of coagulation. And now this thrombin converts fibrinogen into fibrin. Now this fibrin should be lysed into the fibrin degradation products, जिसको हम D-dimers कहते हैं. तो what happens that the kallikrein which is activated and which converts high molecular weight kininogen into bradykinin, and the kallikrein also cause activation of the complement component C5 into C5a, and additionally the kallikrein will cause the conversion of plasminogen into plasmine, and the plasmine will cause degradation of the fibrin into fibrin split products that also play a role in inflammation. So in this slide we can see that the complement system, the clotting system, and the kinine system, they collectively play their roles and are integrated to each other along with the fibrinolytic system to run the inflammatory process. The plasmine also activates C3 into C3a. Now, in clotting system, the Hagman factor are factor 12, and the activated uh, factor 10, it will cause increased vascular permeability and leukocyte immigration, and the intrinsic pathway which results in the formation of thrombin, uh, it will cause increased binding of the thrombin to the receptors on the endothelial cells, and the thrombin will activate, and it will uh, increase the adhesion of the endothelial cells to the vascular endothelium, and thrombin will generate fibrin degradation products जिसको हम D-dimers कहते हैं, that increase vascular permeability and are strongly chemotoxic. Thrombin also cause breakdown of C5 to generate C5a, and in this way thrombin links the coagulation system which is to the complement system, because thrombin is a product of the coagulation system. It is a product of the blood coagulation. But since it converts the complement protein C5 into C5a, so in this way we can say that the two systems are integrated, and the thrombin, it links the coagulation system to the complement activation. Now, the fibrinolytic system, in which the plasmine is the major component, it will cause C3 uh, to its breakdown into C3A, and C3A then causes vasodilation and increased vascular permeability. And plasmine itself can also activate the Hagman factor or factor 12, which is involved in the intrinsic pathway of coagulation. And in this way, the entire set of responses that occurs in inflammation and during blood coagulation, they are coordinated and linked to each other. Summary, in summary, we say that the activation of complement system uh, occurs by microbes, antigen antibody complexes, inflammatory mediators, and it will lead to the breakdown of various complement proteins, and these complement proteins are involved in leukocyte chemotaxis, uh, particularly the C5a, and opsonization, the C3b, and phagocytosis, the membrane attack complex, and opsonization by the C3b, and these components are involved in killing of the bacteria and other microorganisms. In coagulation system, the major component is the Hagman factor, which triggers the clotting, kinine, and complement cascades, and activate also the fibrinolytic system. And the kinines, which are produced by the proteolytic cleavage of the inactive forms, are their precursors, and they, for example, bradykinin is involved in pain and other signs symptoms of the inflammation. Thank you very much.